It makes me a little nervous to speak with all the experts in the audience on CCC DNA, or to even call it CCC DNA with Professor Laccarnini here. I should call it the mini chromosome. I'll do my best here. But we also will find this is going to be the least data-intense topic because there's really not much in clinical development out there on CCC DNA targeting specifically. So what I hope to do today instead is give you a general approach as to how we view clinical development for CCC DNA targeting, potential places we can intervene, and what those effects may be. And I will say some of these topics you'll hear, you heard about already from the last talk, and we'll hear probably in the next talks, because this is the general goal of everybody in the field, is to really affect CCC DNA. So I don't just spend much time on why we're targeting CCC DNA. I think, of course, we're trying to get to the bottom two bullets, which is getting to a functional cure. I think the really stretch goal is to get to the sterilizing cure, but in the intermediate, there's a lot of effects that can be had by targeting CCC DNA that can have effects on new hepatocyte infection, improving the host immunity, reducing integrated HPV DNA. I think those all impact maybe the course of the disease for patients and eventually leading to cure. So important to bring up at the beginning are what are the challenges of targeting CCC DNA, and these are just put into three buckets here. The first was really the host machinery, and I think it's important to note that, and this is, I think, the point that Professor Laccarnini makes, is that the way that cccDNA is regulated is a lot to do with how host genes are regulated and host chromosomes are regulated. And with that overlap of host machinery, it does provide a layer of difficulty with trying to target those processes. So whether it's the epigenetics, the writers, readers, erasers, whether it's the actual transcription and translation from the DNA to the RNA or RNA to protein, or even the DNA repair pathways that go into the rcDNA, the cccDNA transitions, those all likely involve all the same host patterns that your body uses. So those are important things for us in drug development to think about when trying to target those aspects. Again, as been mentioned many times today, the biology of CCC DNA, there's been quite a bit discovered and a lot of new insights from the past couple of years, but there's still some fundamental questions that need more research. And I think understanding those topics will help us in the drug development side guide our therapies to really be most effective. So the mechanisms of CCC DNA formation, we know how it gets into the cell, and we know most of the processes that lead to the deposition of CCC DNA, but there are still some gaps in that, particularly that rcDNA to CCC DNA transition. We heard right just now about the intracellular CCC DNA amplification. I think it's been very well described in other preclinical model systems, and in hepatitis B in patients is now getting more data to show the importance of that pathway, but really understanding how it contributes to the persistence of CCC DNA and then the lack of cure in patients will be important for us to know how important that is to be targeting. As Stefan mentioned as well, I think I'm taking your list here, the half-life of resting of cccDNA in the resting cells and in dividing cells is really important. There's been some recent data to show what may happen to cccDNA in dividing cells, but I think that the resting cell, the post-mitotic cell, is really important to know what the fate of cccDNA in that cell is. And then the mechanisms of silencing. We know that patients who have an acute infection and can achieve a functional cure on their own have some way of silencing their CCC DNA, and I think understanding those mechanisms better might give us better ways to target the virus. And I do want to bring up the integrated DNA, mainly because as we think of targeting specifically CCC DNA, it does become an important factor to realize that there may be components in the host chromosomes of the actual HPV DNA. So in general schematic of how we might approach targeting CCC DNA, we've heard quite a bit already on how we can eliminate the production of CCC DNA in the first place, and that might be the best way to make sure we don't have any more that's formed. Beyond that, there's the amplification, this intracellular process that still needs more definition, but I think that's another way we can get more production of CCC DNA in the nucleus. We can focus on the persistence, so that half-life of CCC DNA. What can we do to destabilize or eliminate the CCC DNA from the nucleus? And then finally, the activity. So if it is present, how can we make it such that it's in a compacted state, non-transcribing, and maybe get to a state of a functional cure? So the effects of targeting are shown on the right, and what I'll do is just go through each one of these with potential ways that the current compounds in development or ideas might fit into those. So first, for production, I don't need to talk more about Mercludex. I think you heard quite a bit already, but I think the goal here is really how do we block new hepatocyte infection. And I think we know that, of course, entry inhibition is a wonderful way to stop new hepatocytes from being infected and prevent the CCC DNA from being formed. I think there's a lot of emerging data from the capsid modulators to show that at high doses, capsid modulators, while not preventing the entry into the cell, but can prevent the deposition of cccDNA or rcDNA into the nucleus. So maybe potentially that's another mechanism to prevent that intrahepatic deposition into the nucleus. And of course, as Stefan also mentioned, other molecules that might block entry, including cyclosporin A, might be other ways that we can work on that. Now, another way to look at it is if you can completely eliminate virion production, you can also essentially achieve capsid or entry inhibition. So nucleotides we believe do not completely block replication. I think we presented posters on that in the past. But how can we combine those with other molecules, potentially capsid modulators or siRNA, other ways to really drive viral replication down to zero? And if you can really do that, potentially you have reduced that ability to infect a new cell and deposit the viral mini-chromosome. And then in that production of cccDNA, it is also a transition step of rcDNA to cccDNA, which likely requires a lot of host factors. But I think that's one other step that you can look at to prevent the actual deposition of the chromosome. So, however, there's a lot of challenges in this as well. I think we need more data for the role of new hepatocyte infection in virally suppressed patients. I think Fabian has gone over some data today also on ongoing replication within the liver in patients who are on nukes. We have data showing that we can still measure peripheral viremia in patients who are suppressed on nukes, but really it's linking that ongoing replication to maintenance of new hepatocyte infection. We still are lacking some of that data, and that would be nice to know if that is really causative for the ongoing presence of cccDNA replenishment. Again, as Stefan pointed out, the intracellular amplification, I think we have some more data on the value of that pathway for maintaining cccDNA, and more data would be important there. And then, of course, as we're using host processes, really the specificity of the rcDNA to cccDNA transition and making sure it's not going to have host targeting effects will be important. So that second avenue is reducing the amplification in the cell. And I think this is much like preventing new hepatocyte infection in terms of things that can be used. And really, again, if we can prevent virion production and DNA amplification in the cell, you can maybe prevent that process of intrahepatic recycling. And nucleoside analogs, capsid modulators, siRNA, all of those still affect here and potentially could have effects at preventing that re-import of rcDNA into the nucleus. And again, I think the main data we need here are really understanding the role of this pathway. So if this one pathway we really want to target, we should really understand the value of it in a chronic infection. And I think the more data that gets generated, the higher yield this kind of pathway becomes for targeting. So moving to targeting persistence. So now we're moving away from the formation and saying, how do we target the persistence of the cccDNA in the nucleus? And I think you can think of, or at least we think of it as maybe some indirect mechanisms and direct mechanisms. So the indirect ones, I think there's many cytokines that have been demonstrated in literature that may impact the stability or potentially even the epigenetics of cccDNA, whether it's through ApoBec or other mechanisms that they can reduce cccDNA either through interferon alpha, gamma, TNF alpha. And I think those are things that many companies are looking at as ways to potentially destabilize and reduce the amount of cccDNA in the nucleus. Cell division, I think, is an interesting avenue as well that Professor Dandri's paper recently really demonstrated that through cell division you can have loss of cccDNA, and that is an indirect way to change the persistence, which is through replication, but I think that's one that needs to be at least considered in the process. And then there's direct effects, direct ways of targeting persistence. And of course, the most direct would be some kind of DNA editing technology, whether there's CRISPR, Cas9 systems, ZFPs, TALENs, meganucleases. There's quite a bit of data in the preclinical setting to show how these can be useful in targeting cccDNA, leading to mutation or elimination of cccDNA and reduced transcription and translation afterwards. Another more slightly direct way for the cell is really trying to target these cells for destruction. And I think you can think of engineered T cells, potentially bispecific antibodies, things you've potentially read about from Drs. Bertoletti and Protzer as well, on how we can try to get that cell to just eliminate the cell and therefore eliminate the cccDNA. Of course, the challenges with both of these approaches are great. One is the efficiency of DNA editing and potential for off-target editing if you're going to go in with a CRISPR-Cas9 or other type of meganuclease. Important to understand also the integrated DNA and what effect that can have if you're going to target DNA editing itself, and what that might mean for chromosomal breaks, et cetera. If you want to take a more T-cell or bispecific antibody approach, really controlling the rate of hepatocyte elimination is critical. And I think that's something that we don't know quite yet how to do, but one that has to be figured out before this can be a viable approach for this. And then finally, targeting cccDNA activity. And I think this is, again, the overlap with host proteins is quite high. And so really thinking of how we affect the epigenetic status, histone modifications, DNA methylation itself, potentially capsid modulation, depending on the data emerging on how capsid's role is in cccDNA chromatization. Other approaches, rather than just looking at the epigenetic state, is trying to target HBX. There's very good data now on the role of HBX in allowing transcription to occur from HBV mini chromosomes. And so if we can find ways to target HBX and then allow this innate SMC56 activity, you can then potentially not target its persistence, but really target the activity of cccDNA. Challenges, as we talked about, is really, I think, till now, an incomplete understanding of the actual epigenetic mechanisms. There's been some nice publications in the past, including this one from Chopberger et al. On PNAS, which described the epigenetics that are associated with HBV, but I think more work really needs to be done to define which of these methyl marks, histone marks, are viable, are really important for the persistence of replication from cccDNA. And as I mentioned, the host machinery really brings another important safety consideration when you're targeting this type of activity. So just briefly, after that kind of overview of how we might approach HBV cure, I just want to take a sense to say how do we think about it at Gilead and maybe other companies as well, is you really can take a biology approach or a phenotype approach, and I just want to point out some of the differences and pros and cons here. So if you take a biology-based approach, we really rely on the fundamental understandings of cccDNA biology that occur usually from academic labs around the world. And the nice part about it is you have a mechanism, you have validated targets you can go after, and I think that makes a very rational approach for trying to target cccDNA. The downsides are that biology is very difficult with cccDNA, and the discoveries happen over a long period of time. Also, those discoveries don't take into account the potential host effects of targeting those regions, and so for us, there's a higher biological risk of having host effects when taking a biology approach. A phenotype-based approach is screening, so that's screening cell lines for inhibitors of cccDNA activity, formation, or persistence. For us, that's nice. You can do rapid evaluation. You can assess various aspects of cccDNA biology from its presence, maintenance, and persistence and activity. And you can immediately select over host toxicity in an in vitro setting, so you can really get to compounds that may have a higher potential. But the real downside is the mechanisms in a phenotype-based approach are unknown. And so that requires us to do a lot of back biology to understand what we're targeting with our compounds. And then we have limitations in the models. So we only have a certain amount of time we can take cccDNA out to in our in vitro systems or in vivo model systems. And those model limitations, I think, also potentially limit the translatability of the findings we have from a phenotype-based approach. So in summary, this is obviously a high-value target. It's either directly or indirectly the effect of most of the compounds everyone's working on in the room. But it's one that really requires us to continue to focus on it. And we, I think, need more information on knowing how it's formed, persists, and the transcription of cccDNA, so understanding that biology will be very important. And we have to really think about, as we take more drastic measures potentially to target cccDNA directly, that we really understand the host effects and potential off-target effects of our molecules for patients who are otherwise doing well on their nukes right now. And finally, really the model systems, the more we improve those model systems, the better chance we have of doing the right phenotype-based approaches to really get to compounds in a more rapid manner. So I think that's it. Hopefully I'm under 10-ish.